The title of this presentation is Non-Local and Delay Reaction Diffusion Equations in Mathematical Immunology. We will begin with a short biological introduction, then we'll describe the model, non-local and delay reaction diffusion equations. We will begin the analysis of this model with non-local reaction diffusion equation without delay. After that, we'll consider delay reaction diffusion equations, and both of them at the same time, non-local and delay equations. Finally, we will briefly discuss some mathematical equations related to the existence of waves and pulses described by such models. Let us begin with a short biological introduction to viral infection and mathematical immunology. Viruses are small particles containing some genetic material surrounded by a protein membrane. Viral infection can concern different tissues and organs. It is shown schematically in the figure at the right. Besides being a pathogen, viruses participate in the process of evolution by horizontal transfer of genetic material among different species. Viruses are also used in genetic engineering. Viruses cannot reproduce themselves. In order to multiply, they need a host organism. When they penetrate the host cell, their DNA or RNA are replicated and new viruses leave the cell. One host cell can reproduce hundreds and thousands of new viruses, and often it dies after that. The host organism responds to virus infection with the immune system. The immune response consists of two main parts, the innate and adaptive. The innate immune response includes cells like macrophages and dendritic cells and interferon produced by infected cells. The adaptive immune response consists of antigen-specific T and B lymphocytes, and their clonal expansion is initiated by antigen-presenting cells. Effector T lymphocytes eliminate infected cells, while B lymphocytes produce antibodies which neutralize viral particles. All these steps of immune response are schematically shown in this slide. The interaction between viral infection and the immune response is schematically shown in this diagram. Viruses replicate in the host cells and initiate the immune response. Viral infection can also downregulate the proliferation of immune cells and promote their apoptosis. On the other hand, immune cells multiply in response to the infection. They eliminate infected cells and free virus particles. Furthermore, we need to take into account spatial distribution of viruses and immune cells, the presence of different organs, and virus mutations. We will consider in this presentation a simplified model for the virus density distribution U of x and t as a function of the space variable x and of time t. The space variable x can have two different meanings. This can be conventional physical space, and in this case, we consider the virus density distribution in a tissue or organ. The space variable x can also correspond to the genotype space. This means that we consider viruses with different genotypes, and uxt is a virus density distribution with respect to its genotype. In this presentation, we will basically consider the second interpretation related to the genotype space. The first term on the right-hand side of this equation describes virus diffusion inside the infected tissue or virus mutations if we consider the space of genotypes. The second term characterizes virus replication in the host cells. Instead of the usual logistic term, we consider the integral term, 1 minus integral, with the integral presented below in the slide. The integral is taken with respect to different genotypes, and it describes the rate of infection of host cells by viruses with different genotypes. Therefore, this term takes into account virus competition for the host cells. Similar integral terms are considered in the models describing the evolution of biological species. The next term describes virus elimination by the immune response. The function f shows the strength of the immune response. Since the immune response is stimulated by the viral infection, it depends on the virus density u, or if we take into account cross-reactivity of the immune response, it depends on the integral s presented below. As before, the integral is taken with respect to different virus genotypes. Furthermore, there is time delay in the immune response due to the duration of clonal expansion of antigen-specific lymphocytes. The last term in this equation describes virus death independent of the immune response, and which can depend on virus genotype. Specific forms of the function f and sigma will be considered below. Though this is a simplified model, it becomes quite complex because of the integral terms and time delay. We begin to study it without the non-local equation, with a non-local equation without time delay. And we'll discuss the questions about virus quasi-species, their existence, competition, and dynamics. Thus, we consider this non-local reaction diffusion equation for the virus density distribution U of X and C with respect to its genotype X. First of all, we are interested by positive stationary solutions of this equation localized around some average genotype and decaying as the distance from this genotype decreases. From the biological point of view, such solutions correspond to virus quasi-species. They correspond to one virus strain or to ensemble of virus strains with close genotypes. Instead of biological species in the case of animals, such solutions characterize quasi-species in the case of viruses. We begin to study the existence of virus quasi-species in the simpler case without the immune response. Hence, we consider virus mutations, reproduction, and genotype-dependent mortality. This situation corresponds to cell cultures, but not to a host organism. The function sigma of x, which characterizes virus mortality rate, is shown in the left figure. We take it for simplicity here as a piecewise constant function. It equals 0 inside some interval of genotype values and to some positive value outside of this interval. This interval with 0 function sigma of x is a viability interval where the virus reproduction rate exceeds its mortality rate. In this particular case, the stationary solution of this equation can be found explicitly. In a more general case, where the function sigma of x is not piecewise constant, the explicit solution cannot be found, and more sophisticated mathematical methods should be used to study the existence of solutions. In particular, we apply for such problems the topological degree theory. I will return to this question at the end of my presentation. Analysis of the existence of solutions of this equation shows that it has a positive stationary solution decaying for large x in the two possible cases. First of all, the diffusion coefficient should be sufficiently small. The diffusion coefficient corresponds here to the virus mutation rate. Therefore, virus quasi-species exist if their mutations are sufficiently slow. This result is in agreement with the biological data. On the other hand, small values of the diffusion coefficient can be transformed by a change of variables to a sufficiently wide viability interval. So virus quasi-species can also exist if this interval is large enough. If there are two different viability intervals where virus replication rate exceeds its mortality, then two different quasi-species can exist, each one in the corresponding interval. Depending on the values of parameters, these quasi-species can coexist at the same time, or one of them can eliminate the other one. The coexistence is shown in the left figure at the top, and the elimination of one quasi-species by another one in the left figure at the bottom. It is interesting that they can coexist asymmetrically, with a high value for one quasi-species and a low value for another one. This case is shown in the middle figure at the left, and the same case is considered in the right figure, where we can see here the evolution in time. After a short transition period, the stationary distribution are established. The competition of virus quasi-species can lead to the emergence of resistant strains due to the antiviral treatment. An example of such simulation is shown in the left figure. In the beginning, there is only one quasi-species, and the second one in another viability interval does not appear because of the competition with the first one. If we apply antiviral treatment and eliminate the first quasi-species, then the second one will emerge because there is no competition anymore, and the treatment does not act on the second quasi-species. This is one of possible mechanisms of the emergence of resistant infections due to the antiviral treatment. A more complex example with 3 quasi-species is shown in the right figure. Up to now, we considered virus quasi-species in the absence of immune response. Let us now consider the second case with the immune response, but without genotype-dependent mortality. As before, we begin with the existence of localized positive solutions which characterize virus quasi-species. The bifurcation of such solutions is shown in the left figure. If the diffusion coefficient is sufficiently small, then the homogeneous in space stationary solution loses its stability, resulting in the emergence of a positive non-homogeneous solution. If we further decrease the diffusion coefficient, this solution becomes more localized, as shown in the right figure. Thus, as before, we find pulse-type solutions corresponding to virus quasi-species. Such solutions exist due to the presence of immune response for sufficiently low mutation rates. Next, we consider the case of genotype-dependent immune response. The function f depends now on the space variable x. This means that virus elimination by the immune system depends on the virus genotype. In this case, we observe in the left figure the evolution of virus quasi-species. They escape the immune response and move to the genotypes for which the immune response is weaker. This result is in agreement with the biological data. A more complex behavior is shown in the right figure. In addition to the motion of virus quasi-species in the genotype space, there is also the emergence of new quasi-species. Finally, we consider the case where both factors, the genotype-dependent mortality and the immune response, are present. As before, we studied the existence of virus quasi-species, their competition, and evolution. This bifurcation diagram shows two different domains in the parameter plane where two quasi-species coexist. It is above the curve, and where one of them eliminates the other one due to the competition between them. This is a domain below the curve. Next, we shortly discuss periodic structures and waves described by non-local reaction diffusion equations. Due to the presence of non-local terms, the homogeneous in space stationary solution can lose its stability, resulting in the emergence of stationary periodic in space solutions. We see here in the figure how a small perturbation of a constant solution grows and the solution converges to the periodic in space solution. The next slide shows the propagation of a periodic wave. There is a periodic structure behind the wave front. There are different regimes of wave propagation. Some of them are shown in this slide. In the left figure, there are two consecutive waves propagating one after another with different speeds and separated by some distance. The first wave is a wave front, and the second wave behind it is a periodic wave. In the simulation shown in the right figure, the periodic wave propagates faster than the wave front. They merge and they form a single periodic wave. In the next part of this presentation, we will consider delay reaction diffusion equations. Time delay appears here due to the duration of clonal expansion of immune cells of the adaptive immune response. We begin with a simple particular case. There is no space variable, and we consider a single delay differential equation. The function f of u has the form shown in these figures at the right. It can be growing, or it can grow for small u and decay for large u. Viral infection stimulates the immune response, so the function is growing for small viral load u, but viral infection can also promote cell death, and it downregulates the immune response for large viral loads. In this case, this function is decreasing for large u. The linear stability analysis shows that the stationary points of this equation can be stable or unstable due to the time delay. In the last case, there is a bifurcation of periodic solutions. Under some conditions on parameters, it is possible to prove the global stability of stationary points. It is an interesting question, but we will not discuss it in more detail now. In the unstable case, behavior of solutions of this delay differential equation can be quite complex. We observe sequences of period doubling bifurcations as it is shown in this figure, and then alternation of periodic and chaotic behavior. Dynamics of solutions becomes more complex if we introduce the space variable. Periodic in time and homogeneous in space-time oscillations can lose their stability with respect to spatial perturbations. In this case, we observe standing waves shown in the left figure, periodic traveling waves in the middle figure. There are also aperiodic spatiotemporal solutions with complex dynamics. One example is shown in the right figure. The upper row of these figures shows a snapshot of solutions. The lower row gives the position of the maxima of solutions on the x-t plane. In the left figure, these maxima are fixed, they don't move. In the middle figure, these are inclined straight lines corresponding to traveling waves. In the right figure, the maxima form a complex aperiodic structure. This slide shows propagation of waves described by the delay reaction diffusion equation. In the upper row, there is a single wave. It can be followed by decay and oscillations as in the left figure, or by periodic oscillations in the right figure. The second row shows the position of the maxima of solutions. There are two consecutive waves in the bottom figures. The first wave propagates faster. It can be a simple wave with a constant solution behind wave front or with periodic oscillations behind it. We conclude this part of the presentation with some examples of simulations of wave propagation in the case where both non-local terms and time delay are present. Depending on parameters, there are various spatial or spatiotemporal structures behind the waves. We have discussed before existence and dynamics of waves and pulses for non-local and delay reaction diffusion equations. In some cases, existence of such solutions can be proved, but it requires quite sophisticated mathematical methods. As I mentioned before, we use the topological degree theory for elliptic problems in unbounded domains and the Leray-Schauder method. To use this method, we need to obtain a priori estimates of solutions in some appropriate function spaces. Some recent publications devoted to the existence of waves and pulses are shown in this slide. In conclusion, we discussed in this presentation non-local and delay reaction diffusion equations arising in mathematical immunology. We studied the existence and dynamics of waves and pulses. They describe the development of viral infection in the organism and virus quasi-species in the space of genotypes. Thank you for your attention.